you. Okay, so now I'm going to talk about RNA sequencing liners and what's so special about them, and what are the different things that you often hear when you talk about these liners. And what's the innovation and what makes them special to DNA mappers or liners are things that I have in common is that they can do read pair alignments. They can consider these base called quality scores. They have created all these sophisticated indexes, so it's possible to map millions of reads in a very short time. The new ones that are out and the one that we are trying has the chance to include genetic variants, so you can increase your mappability and specificity by adding VCF files with genetic variants. And they're resolving multi-mappers using regional read coverage and considering junction annotation. The last thing that sort of the new thing is the two-step approach, which doesn't rely on known annotation for junctions, but basically discovering them and improving the mappability. And I'm going to go through all of these in a bit more detail, so bear with me for the different steps. So most aligners are using seed and extend approach. So in this file, you can see that there are a gene that contains 2 exons. This one over here. I use this nice color. And on the genome, that one is then separated by an intron. So here is an intron. So there's a distance here between these 2 exons. But when the RNA that the reads are coming from is this spliced out form. Okay, so what happens then is that this RNA is converted into cDNA and being fragmented. So now we have the fragments that goes into the library, and that one is then being sequenced, giving reads. And now these reads that are coming out are coming from either exon 2 or from exon 1. But then there are also going to be these reads that contain this part that is from exon 1 and this part that is from exon 2. So what happens then? Well, what the program does is that it takes one of these reads. So here we have a read, and it breaks it up in different smaller parts of a certain length, of a k-mer length. So a k here is a number. On average, that usually 20 nucleotides. So it breaks up the read in 20 nucleotide pieces. And then have created an index with all the different 20 nucleotide sequences of the genome, it can sort of go in and say, I have this nucleotide sequence, where is that located? And then they are going to be identified that these can be mapped to all these different locations. And when you do that for all the different pieces that you have split up, this is in most cases sufficient to give the information where on the genome that read has been mapped. But what it does then after that is that it I don't see my it extends, so it binds together these seeds and creates a full alignment that is then know where it is on the genome. This is so okay. But then there are other reads like this one, which contains information from both these two exons that are separated by an intron. And then for some of these seeds, they are being mapped to the right position on the on this side, and then some of the seeds are being mapped to this side. And now the distance here is too big, so it can't really extend them together, but instead it gets two separate places, two different parts that is split up. And then by using information about how intron-exon junction usually works, it can identify where it's being spliced, where is this intron-exon junction, so it can link them together and identify the intron in between the two exons. So that's basically how you do it. You take your read, you split it up in small pieces, you align these pieces to the genome, and by having these pieces, they can extend them and bring them together. And then the next step, identify where is actually the intron junction and create a mapping like this. Okay. So what is the problem? Well, this should be quite easy, right? But the problem that is facing is that the introns are quite big. So this is a cumulative distribution of the introns and how they are big in the human genome. And there are some that are really small, they are 100. But if you look at the average size, which is here, it's approximately 1,000 nucleotides. But there are also some introns that are as big as 100,000 bases. So you have to allow for these different parts of a read to be mapping very far away from another and map them together. And now, since the signal for identifying what is a good splice site, intron-exon junction or a splice site, is basically only two nucleotides that should be a G and a T, and an A and a G on the 3' end. That's all the information that is available for determining that, okay, this is actually a good splice, as this is where the splice should happen. The programs are having sometimes a hard time to find the correct location.
So we have a question from Jacob.
Okay, you mentioned that the reference we are using contains the information where exons and introns are located. But what about if we have a genomic rearrangement and we have intergenic sequences inside an exon? Is this somehow processed or included?
So I said that there are you can include the information about the annotation of genes in this program to help it to identify these intron-exon junctions. But the reference that you're going to use does not contain that information. If you have enough reads and you have the programs that are working now, I mostly recommend people not to use an annotation at this step, but basically let the reads and the data give that information by using this two-step process. So we're gonna have a look at how that works later. Is that?
Yeah. Thanks.
Good. Good. So just to make it a bit more complicated, there's actually some of the known introns that does not have this GTAG. So that means that the programs that are used does have to allow for other types of intron-exon nucleotide sequences as well, which makes this even more problematic to actually get it at the right location. Yes. Another thing that it has to deal with is this pseudogenes, which is quite occurring in the genome. So basically, you have genes that are, you have pseudogenes that are sort of brought back by reverse transcriptase into the genome again of coding genes that have already been processed. So we move out the introns. And how do you distinguish if it should go to the functional gene location or to one of these processed pseudogenes? And that's not easy to tackle, but the problem, or what it raises, is that the programs can't really penalize opening up for introns when they're mapping, because then we would favor to map to pseudogenes instead of the functional location of the gene. Instead, there's a higher, what is being penalized is, instead, if you are having mismatches or error between the read that you're mapping to the places in the genome. And often, since these pseudogenes have a higher mutation rate, so they are often acquiring more and more errors. So therefore, these functional genes are often being preferred. Having said that, that's one of the reasons why, for example, HiSET2, which is the one that we are working now, allows for adding in genetic variants in the file, so to make sure that you favor the mapping of functional genes compared to pseudogenes. But again, the file that we are going to use today does not contain this extra information about the different variants of mouse. And the reason why you do that is because if you want to do this step by adding this information, you need to have a more stronger computer than your personal laptop. It requires between 100 and 150 gigabytes of RAM storage to process all that information to create a good file. So we don't have that information. But if you think that that could be an issue and you could solve it, you can actually address that by adding that information. So how important is it that you get correct mapping? And that's really dependent on the question that you're asking. And most people, they want to do differential expression analysis. And then the answer is it's not so important because it's the numbers that counts. So even if you get some wrong, since you have so many, it's not an issue that you get some wrong mapping. But then as you get into more specific questions, like you want to identify novel genetic variants, or you want to identify RNA editing, you want to do genome annotation, or you want to do this allele-specific expression, then single nucleotides changes are going to make big differences in some of the outcomes that you're doing. So then it's more important to be very strict and very thorough in how you do the mapping. But for differential expression, which most people do, it's not a big thing. You can basically use any program and you will find some differential expressed genes. So sometimes we get the question which programs to use. And these are some of the ones that are sort of frequently being mentioned. And I'm not going to spend too much time on this. I'm basically saying that the two programs that are mostly used are being HiSAT, which you are going to try today, and the other one is STAR. And the reason why these ones are being used is because they are fast and they produce good results. And then there are other programs that also produce good results, but they're not as fast So it's not really any meaning to use them instead. And this top hat, which is many people are using still, is the same author that did HiSat. So there's actually no reason at all to ever use top hat again. It just takes a lot of time, and it does produce worse results than these ones up here. Okay. So one of the biggest issues that you have, or like if you want to do very good mapping, is this problem of identifying the true intron and exon junctions like this. And the problem when this arises is the cases that you see down here, where you have a read where most of the read is belonging to exon 3, and just a very, very tiny part here, let's say 2 nucleotides or something, are in this location here. So this one is probably a line that you can see, oh, it should be here, but where should we place this one? And since you only have 2, now we only have 2 nucleotides to place somewhere between 100 and 1 million positions from that position. It's quite, it could be in many different potential locations where you can put this. So then the program says, like, I don't know where I'm going to put this. I don't have enough information. So I want to leave those unmapped. So you can't really map those. But what the new programs are doing now, which is this 2-step mapping, is that they are doing a first round of mapping. And then they identify some of these that they don't dare to map, but they find somewhere you have better representations like this. And by using these reads that you could map with high confidence, you can start to identify which are the introns that are represented in this file. And now they can see, okay, there is an intron going from exon 3 to exon 2 here that is represented in the reads that we already have. So what happens if I take this file or this read and I use the same already known intron and take these 2 nucleotides that I have here? Do they have the correct sequence to put it here? And yes, they do. So in the second round, you're now saying with confidence, because there were other reads that identify the intron exon junctions with the introns here, you can say that these reads should also be mapped, and they have only this small fragment here. So you can actually increase the number of reads you map by doing this extra step. And this is what HiSat2 does, and this is what they they're mostly proud of this program compared to other ones, because they sort of came up with an idea how you can do that in a fast way. You can also do the same thing in STAR, but then you have to run it 2 times. Whereas in HiSat, they do this on the fly. So basically, as soon as they start reporting this, they say that this is a known injunction, and then they start adding these extra ones in just one go. So that's the beauty of this one. STAR uses another way, and that's called soft clipping. And that is basically if you're not so it's not so important to get the entire read right. The just important thing is to actually map it to a gene. So then these 2 up here, they can map with confidence, but these ones, there are like some parts that it can't map. They don't know where it is. So instead of just throwing them away, they are just clipping this part of the read away. They say, okay, I'm not gonna care about this part, and not this either. I'm gonna clip it away, which is called soft clipping. And I'm just gonna tell them that we managed to map the read here, but there are actually 2 nucleotides which has been soft clipped and removed that we don't know where to put. And that's fine. And the same thing here, they just remove this part. So this is called soft clipping. And that's another way of handling. And if you are only interested in if you're only interested in, for example, differential expression, this result is just as good as actually having these extra parts aligned. It doesn't add anything to have this perfect mapping. So how good are the programs for mapping? And all programs for mapping RNA are good.
There is a question, sorry, from Jacob.
Does the program tell us that that some of the nucleotides were soft clipped?
Yes.
Okay.
So that comes into the SAM file there. There's it tells you that we soft clipped 3 nucleotides on the sides.
Great. Good.
Okay. So I don't know if you look at mapping accuracy for all these programs.
And sorry, there is a question from Carl.
Yeah, sure, good.
Yeah, sorry, maybe this is still a question. So in the reads, wouldn't you also get some reads that do have introns because they haven't been spliced? So isn't there just a way to kind of correct with that information?
So there will be some reads that contains, you have some intron retention, right? So, but I'm not sure how that would help. So basically, if you have a read with intron retention, basically it's, let's see if I can draw. You have a read that goes like this. Is that what you're meaning?
Yeah, or just reads that are not spliced. I'm just asking this because when I work with single cell data, you can divide the unspliced and the spliced reads. So I wonder that how common is that, and if that, because the ones that are not spliced and you just align to the reference, right?
Mm-hmm. So the ones that are not spliced, yeah, how do you know which ones are spliced? Maybe I'm missing your question here, but you can definitely get the information for which of the reads that were spliced and which ones were not.
Yeah, I'm just wondering, like, if you have if you have reads that contain introns and reads for the same location that don't, then this problem is solved, right? So because then you would map both reads and then you would know, yes, it's actually here, because also the nucleotides that are belonging to the intron would just align to the reference genome, right?
Yeah. So yeah, so I mean, this is I don't know if it solves it, but I mean, the program can definitely handle both of these RNAs at the same time. It doesn't require that you have that. I don't. So maybe I'm missing something, and maybe we can discuss this later. But I don't see the advantage of having the intron parts as well.
Yeah, I'm not saying it's an advantage. It's that I'm wondering if it's something that just commonly happens, and then you don't have this problem.
So so if you have a good representation of a RNA library of coding reads from the cytoplasm, a very small fraction of your reads or your fragments or RNA should contain any introns. But if you have, for example, small RNAs, single cell RNA, or you have other ones where there's a high risk that there has been some integrity loss of the cells, so you have a lot of nucleus RNAs coming out in the sequencing, then you will have more representation of your introns, because you have unspliced reads that are being sequenced. So that's actually a way of validating how good your RNA is. If you find a lot of intron reads in your sample, then you probably have a lot of reads that are coming from the nucleus. And that could be good or that could be bad. It's just that it's something you have to think about when you're analyzing the data. And the biggest problem is, of course, if half of your samples has a lot of reads from the nucleus and half of them don't, because then that's going to be an issue that you have to adjust for. I don't know if that was that answering your question?
Yeah, I think since I'm used to work with single cell data, then I get some unspliced reads.
Yeah.
But I guess if you select for mRNA, then you mostly get just the exons, right? So then I get it.
So like for the, you mean from 10x, I think, right? That you're probably more commonly used. So what you need to do that for align to also introns is to change the reference, right? I don't know if you have run it yourself.
Yeah.
But rather than include the reference that only include the exons, you also include the reference that also has the intron regions, so that allows the program to align to that region as well.
Yep. Mm-hmm.
So that's basically the only difference. And then you have the results without or with the intronic regions, and then that's how you can distinguish one from another.
Okay. Yep.
Okay. So yeah, mapping accuracy is good, independent of what program you're using, especially for differential expression. You don't have to think too much about it. The only thing I want to say is that you should use the same mapper for all your samples, because there are some differences. So you will get different results if you're using different ones. That's the most important thing. But if you remember, STAR and HiSat were much faster than the other ones, and they don't really perform worse in mapping accuracy by any means. If anything, they are doing a better job. So that's why you should use this. I recommend STAR and HiSat. The funny thing is that in this, so this figure is from the HiSat program, where they show that STAR is doing worse than HiSat. But actually all these incorrectly mapped reads are soft clipped reads. And they are mapping just as fine as HiSat if you want to do differential gene expression. It's just that if you want these small anchors to also be mapped correctly, then you will run into this issue, and then you have to address that with doing STAR2 one more time or run HiSat. Yes. Oh. Yeah, and this is also from their paper, and it's just sort of now if you only focus in on the ones where you have a small anchor, so these ones that are hard to map. And this is of course for selling their program, and here you can see how bad STAR gets for these small anchors. They can basically not, in the really hard cases, they can't map anything, whereas HiSat is doing a really good job. Yeah, I think it's a bit false advertisement, but yeah, again, if you want to do genome annotation, or you want to do stuff like that, yeah, I would probably still start with HiSat. Okay, so this is another thing that is sort of this ongoing thing, and I was trying to, in the first lecture in biology, talk about this, like what kind of junctions do you find when you map reads? And then if you actually go in and have a look at what types of intron exon junctions that are being identified when you use the data, and you split them up into things that weren't annotated before and things that has been annotated before. So annotated junctions versus novel junctions, and you see how many reads are supporting the ones that you have, and then you can see that for all the programs there's like 150,000 or so, except for this one, I don't remember which one, but basically the 150,000 of the annotated junctions have a representation of at least one read, and then it doesn't change that much. So almost 140,000 of these annotation has also a representation of 10 reads. So meaning that there are lots of reads that are covering this intron-exon junction, and therefore you can do filtering easily, saying that, okay, I want at least 10 reads to cover one intron-exon junction to believe in it, and you would basically not lose that many of the known annotated correct junctions. But if you look at all the novel junctions that are showing up in the data, and they are like different for different programs, how much some they find 300,000 new ones, some find 100,000 new ones. But then you see that it's a really fast decay on how many reads that are representing these ones. So if you would say, I will only believe new novel junction that has at least 10 reads covering it, you're independent of what program you're using, except these ones, you're going to have a very few new novel junctions coming up. And I think this shows two things. One thing, it shows that the aligning programs are doing a really good job of identifying the true intron-exon junctions, because most of the ones that they find are already been annotated before, which is, I think, impressive. And it also shows something about the RNA sequencing data of the biology, that you're going to have some junctions that are found very rarely, and these might never have been seen before, but they are also most likely non-functional, and it's just basically noise. But they are still sort of being found, but I wouldn't pay too much attention to them. And I think if you're sort of in discussion about that, I think Lucille might have more insight in this than me, but I'm going to leave it at that now. So the input is the sequence reads, and the output is the read mapped to the genome, and that's the SAM. It's a SAM format, which is going to contain the name of the gene, where it's mapped, the scoring. And here you can see this question that was asked. Oops, which is this? So for this specific read, 96 of the nucleotides mapped to the correct location, but there was 5S. So it basically didn't know where to put the 5 last nucleotides, so it just clipped them away and say, this is a soft clipping. So this is a typical result where there's information in the SAM file that it managed to map 96 nucleotides and it soft clipped away 5. Yes. Yeah, so how do you do this? And this is what you're going to try out on the lecture after this is done. So you have done the quality check on the reads. The genome has been indexed, referenced. That has already been done by me. So what you're going to do is to map the reads to the genome, which creates this SAM file. And then you want to convert that into a BAM file, which is basically the same thing as a SAM file, but it's been binarized, so you can't read it by eye anymore, but it's much smaller, so it's comprised, and it's more easily readable for computer programs than the SAM file. So it's more easy to work with, it uses less space, so you should always convert SAM into BAM. And then you do some quality check on the reads. That's what we're going to do tomorrow. And then you have, of course, a chance to visualize the reads mapping on the genome. And we are not going to do this in this course, but if anyone wants to learn or how to do it, you can talk to one of us and we can set that up. There are two that are the mostly used for browsing your results, and one of them is IGV, which you do locally on your computer. And it's has some advantages, and some people are complaining that the interface is not so easy, and it's written in Java. So it has some irregularities. So I don't mind, I like it, but some don't. And they are usually advocating the UCSC genome browser, which is a bit harder to get used to. But once you've done it, there's lots of advantages with it, that you have a lot of public data that you can add in for your analysis, and it's really easy to fast share your data with other persons if you want to have someone collaborate to share it with and talk to about it. Yeah, and this is one example of the UCSC browser where you have this information about the size of the gene, where it's located, and lots of different reads, how they were mapping to this genome. There's information about the gene located there. And you can also then, of course, that's the advantage here, that here you can start adding in other types of information that is available for bioinformatics. So for example, here you can see how well conserved these parts of the regions are in other vertebrates, in 100 vertebrate conservation. And here you can see that in the surrounding area, it's not so well conserved. There's lots of things that are unique for human, but for this specific gene, it's highly conserved. So this is typically one of those regions where you see lots of reads, and you see that it's conserved in other species, and there seems to be some polymorphism and other things, which would make this, it could be a potentially interesting place to investigate further if you would suspect for some reason that this is a gene that you're interested in. So last slide. So use star or HiSat because they are the fastest. The advantage with HiSat2 is that it uses the least memory of the two, and that's why we're using it today. Otherwise, when I use a cluster, I prefer star, but they are both doing a good job. So if you want to have a perfect mapping, consider these two paths of read mapping. And if you are really important to get it right for HiSat2, you can add SNP data, so you can have even better mapping. If you have longer reads, you can use star, and you shouldn't, if you find some novel introns and they are only supported by some single reads, they are most likely just noise. So I wouldn't spend more time on those doing future analysis. And yeah, it's always good to check the result in some way, and it's always one of those ways is, of course, to use the browser to actually look at the reads. That, for some reason, is actually a really good way of checking your results. Yes, that was all